Welcome to this explainer. Today, we're diving straight into an absolute marvel of biological engineering, dicotyledonous leaves. You know, these aren't just simple green things hanging off branches. Oh no. They are highly specialized, incredibly efficient organic machines that are literally perfectly optimized for photosynthesis.
Think about this for a second. How does one single leaf sustain a small animal? Well, did you know that a well-optimized leaf can actually produce enough oxygen in just 24 hours to keep a small creature alive? It's a staggering statistic, right? It really proves just how powerful these biological solar panels are. And understanding the nuts and bolts of this design isn't just fascinating, it's absolutely crucial if you're aiming to dominate your O-level biology exams and really build that rock-solid foundation for plant nutrition.
All right, so here is our roadmap for today. We're going to cover the anatomy of a solar panel, check out external leaf adaptations, slice into the microscopic internal structures, break down the photosynthesis equation, and finally, help you dodge some common exam pitfalls. Okay, part 1, anatomy of a solar panel. We're talking engineering for maximum efficiency. Moving right along to section 2, external leaf adaptations at the macro level. To really get this, let's just imagine the plant is an elite engineer, okay? And their main problem is maximizing solar capture while ruthlessly minimizing the loss of vital water. First up, we have the lamina, or the leaf blade. Notice those downward yellow arrows? That's the sunlight hitting this broad, flat surface with those dimension lines spanning the entire width. This specific broad shape is literally perfectly optimized to maximize the surface area for grabbing light. It acts exactly like a sprawling solar panel. Plus, this thin, flat design actually speeds up gas exchange and water transport across the whole thing. Now, check out that clear boundary layer overlaid on top of the leaf. See those blue arrows pointing up? That represents water vapor trying to evaporate. But watch, they hit that boundary and bounce right back down. Yet, the yellow sunlight arrows, they pass right through without a problem. This is a brilliant solution to water loss. It's the transparent cuticle. It acts like a literal clear umbrella, trapping moisture inside without blocking the critical light energy needed to power the whole factory. And you know, even their physical arrangement is super strategic. Looking at this from the top down, those scale indicators highlight the precise angles of the leaves around the stem. They're deliberately positioned to avoid shadowing each other. This minimal overlap guarantees maximum light exposure across the entire plant. Basically, no energy is left on the table. Section 3, microscopic internal structures. It's time to slice this leaf open. All right, let's look under the hood. Zooming in first, we hit the epidermis, that top translucent layer. This is your protective outer boundary. And just like the cuticle sitting right above it, these epidermal cells are completely transparent. The sunlight passes straight through them, allowing that light to penetrate way deeper into the leaf, where the real magic actually happens. Now, if we look closely at the lower epidermis, you'll see the leaf's dynamic security system, the stomata and guard cells. Look at the mechanical action going on here. We've got two bean-shaped guard cells, and you can see them swelling up to open that central pore, the stoma, and then shrinking to close it back up. This physical mechanism perfectly regulates what comes in, like carbon dioxide, and what gets out, like oxygen and water vapor. So right in the middle of the leaf, the mesophyll, we have a massive structural split. Take a look at the palisade mesophyll. Notice how tightly packed those elongated cells are? They are absolutely crammed side by side with little green dots. Those are the chloroplasts. This is the main site of photosynthesis. It does the heavy lifting for energy. But then, look over at the spongy mesophyll. See all those vast empty white areas between the cells? Those are air spaces. And those animated arrows weaving through them show exactly how this open layout facilitates incredibly rapid gas transport throughout the leaf. Down at the structural core, we've got the vascular bundles. Think of these as the city's plumbing. Those blue pipelines are the xylem, bringing water and vital nutrients straight up into the leaf from the roots. And the warm orange pipelines, that's the phloem, flushing the newly manufactured sugars right back out to feed the rest of the plant. It's a completely flawless supply and distribution network. Moving to section 4, the photosynthesis equation. Let's look at the chemical flowchart. Following this step-by-step flowchart, we start in step 1 with our raw inputs. We take 6 molecules of carbon dioxide, which we literally just saw diffuse through the stomata, plus 6 molecules of water, delivered fresh from those xylem pipelines. Moving to step 2, we add light energy. This is our ultimate catalyst, captured by those packed palisade chloroplasts. And bam, step 3 gives us the yield.
C6H12O6 are energy-rich glucose plus 6 molecules of oxygen released safely back into the air. That right there is the fundamental equation of life on Earth, mapped directly back to the physical plumbing we just explored.
All right, Section 5, Common Exam Pitfalls. Let's make sure you're avoiding these traps.
You know, students lose marks on leaf anatomy questions all the time because of just a few easily avoidable mistakes. The absolute #1 error? Confusing the roles of the two mesophyll layers. Notice those big red warning markers? If your exam asks you to label the primary site of photosynthesis, you absolutely must point to the dense top layer, the palisade layer, because of its high chloroplast concentration. But if you're asked about gas diffusion, point to the airy bottom layer, the spongy layer. Seriously, do not mix these up on your labels.
And here is a very strict rule for your exam diagrams, beautifully shown in this cross-section. Standard epidermal cells, that top and bottom boundary we looked at, are completely clear. They do not have chloroplasts. The only cells within that entire outer epidermal layer that contain chloroplasts are the guard cells. If you start drawing green dots inside your standard epidermis cells on a test, you are going to lose points. Keep them clear. Keep them transparent.
Okay, let's do a super quick recap of our biological blueprint. We saw how external adaptations like the broad lamina and that transparent cuticle maximize light capture. We sliced the leaf open to reveal those specialized key layers, the protective epidermis, the dynamic guard cells, the dual-purpose mesophyll layers, and the vascular transport pipelines. And finally, we tracked that essential 3-step chemical equation, turning water, carbon dioxide, and light into glucose and oxygen. So, always label your exam diagrams clearly and make sure you state that photosynthesis equation completely accurately. But before we wrap up this explainer, I want to leave you with a final thought. If a single natural dicotyledonous leaf is this efficient at sustaining life on its own, what is the ultimate limit of a plant's growth if we were to step in and artificially alter or enhance those inputs? I mean, how far could we push this incredible biological engineering? Keep that in mind as you continue your studies. Thanks so much for joining me today, and I'll catch you next time.